hi, everybody. For those of you who don't know me, I'm Clark Barrett. I'm currently the director of the Center for Behavior, Evolution, and Culture, and I just wanted to say a few words before we start, given that this is the last event in our speaker series for this year. What I most wanted to say is to give a deep thanks to Susan Perry for organizing the series this year. On behalf of the center, on behalf of all of us, Susan, you've really done a great job. This is our first post-pandemic year, and so there's been a lot of uncertainty about like how to run in-person seminars, and it's been a really nice combination of in-person events and online events as well. So everybody, please join me in giving a round of applause virtually for Susan. Thank you so much. Yeah, thank you very much, and thanks to everyone for being here at this year's speaker series. I just also wanted to say that stay tuned for next year. Our organizers will be Professors Abby Bigham and Molly Fox in biological anthropology. We're going to be they're in charge of organizing the actual speaker series lineup, and we'll be working on that over the summer. I would say if you have suggestions for potential speakers, I would say feel free to. I'm speaking on behalf of Abby and Molly. I don't know if either of them are here, but send them an email if you have an idea, just so that people know. I mean, it's always useful to get a bunch of suggestions for potential speakers up front, so that Abby and Molly can be organizing this over the summer. But I would also encourage people, there's no guarantee that we'll be able to host any particular person if the schedule is full and if it's impossible to find a slot. But really at any point, if you become aware of someone coming through town, or if you become aware of a local speaker who might be interested in speaking, there's really no harm in reaching out to the series organizers and saying, hey, I talked to this person and they I met someone at a conference, or I came across someone's research and they seem interesting. Maybe they could be a potential Beck speaker. I would say, always have that in mind and reach out to the speakers. But certainly if you do have people that that you'd like to see next year, email our organizers. Another thing that I would say is, let's try to also aim for a very, I think as we go forward, it's important to keep in mind that behavior, evolution, and culture covers an incredibly broad scope of possible topics. And so I would say think broadly, think outside the box about people who might be relevant speakers. Personally, for me, I think it's important for evolutionary science to also hear from people who don't have evolution as a main part of their research, if it seems that it might be relevant for evolutionary work. And so, keep that in mind. We want to have a diverse and interesting lineup of speakers. So thanks in advance to Abby and Molly for organizing next year. We're planning on having it be mostly in person, but we'll see. There may also be some room for hybrid talks as well. The last thing that I'll say is that we will also be having as we had this year, the Frank Marlowe Memorial Lecture. As you know, this year it was Professor Kim Hill from Arizona State University, and that was a great lecture. That is also something where we would like to solicit suggestions from the Beck community. If people are interested in suggesting or nominating possible Marlowe speakers, feel free to. Probably the easiest thing is just to communicate that with me, and I'll share it with. We're forming a committee that will be selecting the Marlowe speaker for next year. So do that. And also stay tuned for, we're hoping to have 1 or 2 maybe special events next year. We're not sure exactly what that might look like, but stay tuned. And we're really hoping to continue to kind of keep Beck a vital and vibrant community of scholars working at the intersection of behavior, evolution, and culture. And so we're hoping to have a couple more in-person things next year to keep, to just keep our community active and getting together. So that's also thanks for funders as always, and thanks to you all. Thanks again to Susan, and thank you, Laurel, in advance for coming to speak to us, and we're looking forward to your talk. And I hope you all have a great summer. Thanks.
And yes, thank you, Laurel, for talking. Her talk will be called Cultural Rescue, Avoiding Extinction with Gene Culture Coevolution. Laurel is authored some of my very favorite modeling papers over the years as I study evolution of social learning mechanisms and things like that, and especially the relationship between that and life history. So I'm really excited to hear this latest chapter in her work. And with no further ado, please go ahead and share your slides.
Let me see if I can correctly share the correct thing. Okay, you can see just the slide, right?
Yeah.
Okay, awesome. Thank you so much. Thank you so much for inviting me to be here. I'm super excited. We had a few blips on the way to getting here, a bit of COVID, a bit of strike action, sort of getting in our way. So I'm super glad to be here today to be able to present this to you. So before I kind of dive in, I wanted to flag first that everything I'm presenting today is very much a work in progress. It's also not been in progress for all that long. But all of that is just to say that I'm totally open to, if you have any ideas for, any constructive criticism, any interesting new directions that might make the project more complete or more interesting to you, or any literature I haven't paid enough attention to, I would love to get all of your feedback on any of those things. So I'm totally open to any feedback that you have to give me, and I appreciate the opportunity to present this. Okay, so I am a senior scientist in the Max Planck Institute for Evolutionary Anthropology, where along with Anna Candler, who I think gave a Beck seminar at some point, maybe this year, I run the Theory in Cultural Evolution Lab. So the lab has a couple of aims. So the first and probably the biggest aim is to expand and kind of fortify the theoretical foundations of cultural evolution. And this means engaging in a lot of pure theory projects. And this is what I spend most of my time doing. So most of my work is in purely theoretical projects. The lab also has another aim, and that is to provide kind of statistical frameworks in which to use theoretical models, or at least the insights from theoretical models, to apply to data analysis. So this involves like a lot of projects that connect data to theory. I don't often engage with those projects, but the one that I'm going to present today sort of spans both of those aims. So the project that I'm going to present today aims to understand in a very kind of mechanistic way how genes and culture interact. So particularly gene-culture coevolutionary dynamics in a variety of systems and a variety of evolutionary scenarios. So what exactly do those interactions look like? And then when we go into the genome to look for them, what should we be looking for? So I'm not going to focus a whole lot on methods. I'm sure that there are some people who are super interested in the models. And if you're really interested in the models, I'm going to touch on the structure of the models. I'm going to present one analytical model and one simulation model of kind of very similar systems. But if you want more details on the models than I provide here, you can feel free to ask me at the end. And if that's not enough time or whatever, I'd be totally happy to pass on a draft when it's in some sort of readable shape, which shouldn't be too far from the present, hopefully. So please feel free to ask or to email later on if you would like any more details than I provide here. Okay, so I'm just going to dive in. So the idea here, and the idea behind a lot of the work that I do, is that humans are kind of weird, and we're weird in a number of ways. I think everyone here would probably agree that humans are a bit strange, even among primates. And it's not just that we have weird life histories, and it's not just that we have weird brains, but actually our evolutionary processes themselves are a little bit strange. So it's likely that human evolution involves both cultural inheritance and ecological inheritance and other modes of non-genetic inheritance of a scope and on a scale that's kind of unmatched in other species. So to the extent that we don't fully understand cultural inheritance and the interaction between cultural, ecological, and genetic inheritance, there are huge gaps in our theoretical understanding of evolutionary processes, particularly those ones that pertain specifically to humans. There's loads of historical reasons why that might be a good idea, and loads of people who want to study non-human evolution, and that's totally fine. But if we want to understand human evolution, we need to build a little more theory that deals with the interaction between genes and culture, genes and ecological inheritance, etc. Okay, So most relevant for this project, recent human evolutionary history seems to have been dominated, at least to some extent, by interactions among climate change, demography, cultural evolution, cultural innovations, and genetic adaptation. And we have some evidence to suggest that recent climate fluctuations, maybe since the last glacial maximum or whatever, were attended by expansions and contractions, local extinctions in human populations. So these large-scale demographic changes in human populations. And this raises an interesting possibility, and that is that the demographic and cultural changes that accompany the adaptation of human populations to these kinds of volatile environmental conditions might considerably complicate the mechanics of genetic adaptation, but we don't fully know in what way. So one of the outcomes might be that if we were to look at human genetic data, human genomic data, for signatures of some types of evolutionary processes, we might not find them. And we might not find them because they're not there, completely valid conclusion to draw, or we might not find them because we're not looking for quite the right thing. So I'm not saying that's what happened in this paper. This is a paper that went into the human genome and looked for signatures of classic selective sweeps and found that they were rare in recent human evolution. I'm not saying that that's what happened here. This is just a very cool paper that's related and sort of inspired my thinking on this question. But it's worth reading if you're interested in this kind of question. So this really begs the question, what does, for example, cultural interference in the human genome actually look like when you look at human genetic data? And so this project aims to describe what we might see in the human genome if selective sweeps and complex demographic processes were influenced by the presence of relevant cultural traits. So given that these processes have taken place in the presence of phenotypically and evolutionarily relevant cultural traits, what changes in the signatures of these things could we expect to see? Okay, so because we wanted to look at the interaction of all of these things, so environmental fluctuations, complex demography, cultural evolution, and genetic evolution, we chose to focus on population rescues, which is a particular evolutionary process which entails all of those 4 components, or entails 3 of those components, and we're going to add culture. Okay, so for anyone who isn't really familiar with population rescues, you can probably imagine what a population rescue is, but I'm going to tell you what it is anyway. So population rescues happen when adaptive evolutionary change restores a positive growth rate to declining populations and prevents extinction or extirpation, so local extinctions, after rapid or detrimental environmental change. And this has kind of well-described, well-understood population-level demographic patterns. And there have been a lot of, for example, experimental studies done on population rescues in a variety of species. This arrow is here to remind me to tell you that most of those species are invertebrates. So for example, flower beetles, there are a couple of nice studies, but also in viral populations and in bacterial populations. And if you want to know a little bit more about sort of larger animals, so vertebrates in particular, or mammals, there's a paper by Van der Wal et al. From 2012, which deals specifically with population rescues in these sort of larger animals. Okay, so there are a few types of population rescues that can avert an extinction event in the presence of a sort of very rapid, detrimental, changing environment. So these are 3 that are particularly interesting. So the first is a true kind of evolutionary rescue. So this depends on the emergence of new beneficial mutations for adaptation and for population recovery. So in this scenario, a new mutation arises, and that mutation is beneficial enough to restore the population's growth rate. And the other 2, so demographic and genetic rescue, rely on migration of new individuals. So demographic rescue is where new individuals migrate in, and just the restoration of the population size buffers against stochastic fluctuations and allele effects, and so saves the population in that way, or helps the population to recover in that way. Genetic rescue also relies on migration, but here the new migrants bring with them genetic variation. So they prevent things like inbreeding, depression, and so they can help the population to recover in that way. I'm not going to focus on the ones where migration is important here. I'm just going to focus on evolutionary rescue, so the emergence of new beneficial mutations for adaptation. So we can talk about what the demographic patterns that we see with population rescues, and particularly evolutionary rescues, look like. So here on the x-axis is time, going off to infinity. And on the y-axis is population size. So I've settled on a population size of 1,000. This is completely arbitrary. So my population is going to start out at a size of 1,000 individuals. And it's just going to fluctuate around 1,000, some sort of carrying capacity there for some length of time before the population experiences a kind of lethal, rapid environmental shock. This causes the population growth rate to rapidly decline and the population size to rapidly decline as well. So the population size declines, and then in the absence of any evolutionary compensation or any evolutionary change in response to that, the population will just die out at some time shortly after the environmental shock. But something else could happen instead. So what else could happen instead is that the population might begin to decline at the same rate, but then a beneficial mutation arises. This beneficial mutation might start to spread quite rapidly through the population in a selective sweep through the population. And instead of dying out, the population size might continue to die out for a little while, but then begin to recover, and its population growth rate will recover a high value. And you can also track the frequency of the beneficial allele as it goes from a frequency of 1/n if it's a single mutant, all the way up to, in this case, fixation. So it goes to a frequency of 1. So this is a kind of classic evolutionary rescue. But what I want to draw attention to is that there's this very important period of time between when the environmental shock happens to the population and when the mutation arises. So this is known as the waiting time for mutation. And of course, what the waiting time is depends on mutation rates and on the rate of decline of the population. In many cases, waiting times will just be too long. So for example, when there are long generation times, small population sizes, low germline mutation rates, evolutionary rescues like this are very unlikely to occur in populations. So particularly vertebrates and especially large-bodied mammals. And what you might notice about all of these criteria is that they all apply to humans. So we have very long generation times. We're large-bodied vertebrate mammals. We have low germline mutation rates even compared to our close relatives. So all of this is to say that, a priori we can make a very educated guess that this kind of process isn't going to be very frequent in human evolution. But then we have to ask the question, well, how would a human population really adapt to an environmental shock? So if you think about an environmental shock like the onset of an ice age or a cold snap or something climatic like that, maybe a human population would not adapt genetically first, maybe they would adapt culturally first. So maybe the first thing that would happen is that the human population might invent better cold weather clothing, for example. So the possibility exists that humans will adapt behaviorally or culturally long before they adapt genetically to any of these detrimental environmental changes. So we can take a look at what a process like that might look like. So again, we're back to our population of 1,000 individuals. They experience a shock, and now the population begins to decline. But instead of waiting for mutations, beneficial mutations, or new alleles to arise, instead we wait for an innovation. Now, the innovation may not be able to rescue the population completely, but maybe instead of the population dying out like this, instead it might die out at a much slower rate like this. And that gives the population some breathing room. So the population now has much more time in which a beneficial mutation can arise. So a beneficial mutation might arise over here, where the population might have died out in the absence of a cultural trait, but it's able to be around to experience this mutation. And then we can track the sweep through the population of the beneficial innovation and the sweep through the population of the beneficial allele and the subsequent rescue. Okay, so this is the kind of dynamic that we're interested in modeling. So the first question is, can we generate a model of this kind of gene culture, coevolutionary rescue? And what can that tell us about the dynamics of human adaptation? So we took two approaches with kind of two different aims. So the first model that we wrote was an analytical model. So this can help us to get some expectations for the probability that rescues like this can happen, and to compare this probability to a gene-only evolutionary rescue, all else being equal. And this analytical model should also allow us, if we analyze it in the right way, to get a handle on the mechanics of the interactions between the alleles and the cultural traits as they either sweep to fixation or die out. So either in a population of stable size or in a population that's undergoing an evolutionary rescue. And we should be able to compare those things to understand them a little bit better. And the second thing that we did was to construct an agent-based simulation of almost exactly the same dynamics. And I'm going to talk about the dynamics in a few minutes. And when I do talk about those, they pertain to the analytical model and to the simulation model. But it's important to remember that rescues are very rare events. They happen in small, declining populations, and they're, relatively unlikely to happen. And this means that their dynamics are very, very susceptible to stochastic fluctuations. So in order to get a proper handle on stochastic fluctuations and how those kinds of fluctuations might affect any signatures of rescues that we might be able to find in, for example, genetic data, we need a stochastic simulation to do that properly. So we did that as well. Okay, so I'm going to talk about the analytical model first, and this is also population dynamics that will play into the simulation model. So we start with a population of individuals, and they're characterized by phenogenotypes. So this is like pretty standard cultural evolutionary fare. So it's a genotype that's represented here by this diploid locus, big A, and the ancestral form or the ancestral allele is little a. So this is a diploid ancestral individual. They also have a phenotype or a cultural phenotype, which behaves essentially as a haploid locus. The ancestral form of that is little b. And we assume that the ancestral type has a low fitness, which we denote by W, with a subscript denoting its phenogenotype in this new environment. And we can imagine then if we allow the individuals in the population to mutate and to innovate, they might come up with different phenogenotypes that might look a little bit like this. So here we've got a big A, big B individual. And this individual has big A, which is a beneficial mutation, and they've got 2 copies of that. And they have big B, which is a beneficial innovation, which affects their fitness, W, big A, big B. And we can imagine all of the possible combinations of phenogenotypes that might exist in the population, and we can construct fitness equations to describe their biological fitness in the population. So that would look a little like this. So there are 6 possible phenogenotypes. And we say, firstly, that the ancestral state, by assumption, is the worst fitness-wise in the population. So these guys just have a fitness of 1 minus D, and D is denoting the severity of the environmental shock. So this is how quickly the population will decline. And we also assume that all the individuals are actually suffering from a mismatch between the environment and the genetic and cultural background. So each individual has this 1 minus D detriment. And the model is really about how the individuals with the different genotypes and different cultural phenotypes compensate for D in their fitness. So here, the big A allele confers a benefit HSA, where H determines the effect of a single allele as opposed to having 2 mutations, 2 big A alleles. Typically, H is equal to 0.5. I'm not going to show any results for anything other than H is equal to 0.5. So we say then that 2 copies of the new big A allele confers typically greater benefit, SA. And we can do the same for culture. So now big B is a beneficial new mutation and it confers a biological fitness benefit, SB, to individuals that carry it. And we can do exactly the same as we did before. And that way we construct these fitness equations for every individual in the population. So just as a side note, if anyone's wondering why these equations have the structure that they do, so typically these are done multiplicatively. So you could do this additively and just add these things all up in a row. It doesn't make a huge amount of difference when SA and SB are quite small. They're more or less the same if you do it multiplicatively or you do it additively. But there's a lot of previous work. So the reason that I chose to do it in this formulation is that there's a lot of previous work that pertains particularly to selective sweeps, so Otto and Whitlock, and another model of evolutionary rescue, which I'm going to come back to later, which was by Osmond and Coop, which do it in this multiplicative framework. So I wanted our models to be directly comparable with those ones. So that's one of the reasons why we stuck with this multiplicative framework. Okay, so we have our individuals, and now we're going to put them in a population. So just to demonstrate how the sort of life cycle happens, I'm going to pull apart the phenotype. So I'm going to put the genotype on top, and I'm going to put the phenotype, cultural phenotype on the bottom, and then I'm going to put these individuals into a population. So these individuals are all in a population. Each of them has a fitness benefit or a fitness function associated with them, and we allow the population to reproduce. Now, the fitness functions, of course, as we just saw, depend on SA, SB, D, and H. And if D has a positive value, the population size is likely to decrease in the next generation. After biological reproduction, we have cultural reproduction, and we do that with a success-biased cultural transmission, and the strength of the success bias is determined by this parameter, which we've labeled G. Now, this is oblique transmission. We do have a very similar model with vertical transmission, but I'm going to focus on oblique transmission today. But if you're interested in vertical transmission, you can ask me about it. Yeah. The results are very, very similar. Okay, so G is determining the strength of the cultural transmission bias. And then this just repeats and repeats and repeats until the population either dies out or recovers. So it just keeps going and going. Okay, so once we have this model, what can we do with it is a good question. So the first thing that we can do is to calculate the average biological fitness. So this is W bar, and the average cultural fitness, this is V bar. And we can calculate the change over a single generation in the frequency of the beneficial allele, given that it exists. So we can do some numerical simulations. We can look at the time course of allele frequency change over a selective sweep and over a rescue. So over a selective sweep in a population of a fixed size and a rescue where the population is declining and then increasing. And we can do the same thing to change the frequency of, for the change in frequency of a beneficial innovation. Again, we can look at these numerical simulations, and we can use these numerical simulations to understand how the genetic and cultural traits affect one another as they sweep through the population. So that's the first result that I want to present to you today. So these are the results for allele frequency over time. So this is for the red lines, we're looking at sweeps in populations of fixed size. For the black lines, we're looking at rescues. So these populations have declined and then increased again. And what I really want to pull out of this is that if you look at the difference between a gene culture sweep and a genetic sweep, the genetic sweep alone is faster. And if you look at a gene culture rescue and a genetic rescue, the genetic rescue on its own is faster. So the allele frequency increases much more rapidly in a purely genetic sweep and in a purely genetic rescue as compared to the gene culture case. And this is a pattern that we also see in the cultural trait frequency. So taken together, this kind of shows that gene culture rescues and sweeps in general are slower than either cultural rescues alone or genetic rescues alone. So genes and culture do interact with one another, but kind of contrary to at least my intuition, they tend to slow each other down. But why do they do that? So there's two reasons. One is quite technical, and which I don't want to spend too much time on, but we have to, when we construct these equations, we have to correct for stochasticity in the system that pulls the system away from, that pulls the result of the analytical model away from what you would find in a natural population. So there's a correction that we apply that's based on the probability of the sweep happening, or the probability of the sweep or the rescue being successful. And so gene culture sweeps are more likely to be successful. And so the correction that we make to the initial conditions is weaker. But I think a more interesting reason why this happens is that there is some direct interference between culture and genes. So in the gene culture case, the sweeps are slower, for example, because benefits accrue to little a, so ancestral genetic types with a good cultural trait, who would be completely disadvantaged in gene-only sweeps. So the culture is holding back the spread of the big A allele by providing advantages to these ancestral types that wouldn't otherwise exist. Okay, so what else can we do? So the next thing that we can do with the model is to ask it how long it should take for a cultural trait to fix in the population, followed by how long it should take for a beneficial mutation or allele to arise and establish in the population. And we can use these things all together to generate a probability for the establishment of a mutation and an innovation before extinction. So given a particular severity of environmental shock, which determines the rate of population decline, how likely are we to be able to rescue the population, or how likely is the population to rescue itself before the population goes extinct or functionally extinct? So we put all of this together to generate the probability for the emergence and establishment of an innovation and a mutation, a gene culture coevolutionary rescue, and hopefully also learn something about its dynamics on the way. So I'm going to commit a cardinal sin of presenting mathematical models. And I'm going to show an equation that I'm not going to fully explain. If anyone's interested in a little more information about this, I can definitely give it to you. But I want to draw a couple of things out of this equation. So this is the probability of a rescue. It's the full probability, which includes waiting times and innovations and the effect of culture on the decline of the population, and all of those things in together. So this is as a function of the time to extinction when a cultural trait can arise. And it's made up of predominantly two parts. So it has the probability that a beneficial allele can arise and establish before the beneficial cultural trait. And this is typically quite a small number. So this is typically quite a small probability. And this is typically what most models of evolutionary rescue would entail. And it also contains this part, which is the probability that a beneficial allele arises and establishes after a beneficial cultural trait. So what you'll notice is that where we've applied the environmental detriment, so where we've applied the decline of the population, and we've adjusted it with the benefit of the cultural trait here. So it changes the rate of decline of the population, and it gives us, this is where the breathing room comes from in the equation. So we can use this equation to take a look at how likely are we to get population rescues. This thing depends on G, so it depends on the strength of the success bias in the cultural transmission process. It depends on SA, SB, so these are the biological benefits. It depends on the initial size of the population and the rate of decline, which is D. Okay, so I'm going to show you some of the results from this equation. So along the x-axis here, we have the environmental mismatch, which is D. And on the y-axis here, we have the probability of rescue before extinction. So this is just coming directly out of the equation that I just showed you. We tried to do a few things here. Oh, okay. So the first line that I'm going to show you is So we tried to go into the literature as much as possible and find estimates for example, mutation rates that were sort of comparable with estimates for mutation rates for human germline mutations. So we tried to do that. We took a few estimates and we looked at all of the estimates. I'm going to show you one today, but they were all more or less the same with this parameter combination. And then I'm going to show you what happens in the absence of culture and then in the presence of cultural traits which are transmitted with different strengths of success bias. I've stacked the deck here a little bit in favor of culture. So what I've said is that when the environmental mismatch is down here at 0.05, if the cultural trait appears, it can completely compensate for the environmental detriment. So what that means is that all the lines are going to start up here at 1 because the populations, once the cultural trait exists and spreads in the population can survive for infinity. So we'll always get a rescue. But then you can kind of see that decline over time or over the environmental range. Okay, so you may not have seen much change there, but I actually put the line on for the probability of rescue before extinction. It is this brown dashed line across the bottom. It looks like it's more or less 0, which is kind of what we might expect. It's not actually 0, it's slightly above 0, but for this parameter range that we have chosen ourselves, essentially the probability of an evolutionary rescue before extinction in this case is more or less 0. We can add all of the cultural models on top of this. So even if we look at something like this red line here, where there is not particularly strong success bias, the cultural trait is not spreading that fast, we can still, get a reasonable increase in the probability of rescue from nearly 0 to something more like 0.1, 15% or something like that. It's not negligible. So one other kind of interesting tidbit that came out of analyzing this analytical model was that, so it's worth kind of remembering that we're not just waiting for any mutation to arise, we're waiting for a mutation that's good enough to rescue the population to arise. So the mutation that arises has to be capable of compensating for D to restore population growth rate to a value above 1. So we can calculate these thresholds. So in the absence of culture in our model, we've got this this is the fitness benefit of the cultural trait across the x-axis here and the required allele fitness. So this threshold allele fitness to facilitate a rescue on the y-axis. So this black dashed line shows us what that threshold would be in the absence of culture. And then as the fitness benefit of the cultural trait increases, we get this linear decrease in the threshold value of the required allele fitness. So what this tells us is that culture helps rescues along as we thought that it might, but it actually does it in 2 ways. So first we have this breathing space, so longer to wait for this mutation to arise. And when the mutation does arise, it doesn't have to be as good in order to rescue the population, provided the cultural trait is stable. So all of this is provided that the cultural transmission process is relatively stable. Okay. Does anyone have any questions on that? Because I'm going to move on and talk about the simulations next. No, that's okay. Okay, so so what I want to move on to talk about is more of the stochastic simulation kind of work, which is where we can really bridge between the theoretical insights that we've got and maybe some data analysis in the future. So this is kind of a pipe dream of how we might apply these insights to actual genetic analysis. This is, ideally where we would love the project to go. So what we want to ask is, how can we detect selective sweeps and evolutionary rescues in genetic data? And can we do the same thing for gene culture rescues? Is there any diagnostic differences between gene-only rescues, gene-only sweeps, and gene culture rescues and gene culture sweeps that we can leverage to create a statistical test of sorts. So there are methods, of course, to detect selective sweeps and to detect population bottlenecks. So a lot of them involve detecting kind of persistent changes in heterozygosity, so in pairwise distance or differences between individuals if you're considering a single locus, or detecting persistent changes in allele frequency distribution. So there are loads of different methods, and these are the two that I'm going to focus on today. And we focus primarily on heterozygosity for now. It fits our model. Also, it's kind of a nice, easy measure to work with. But we do hope to develop better methods, maybe based on frequency distributions, so allele frequency distributions and cultural trait frequency distributions. And because, as you'll see very soon, there are some serious issues with the approaches based on heterozygosity, unfortunately. So I do want to say that for a lot of this work, the analytical models and also the simulation models that I'm going to talk about today, or at least the ideas behind them, I relied a lot on this paper, which I really love. And it's by, I guess, your colleagues, neighbors at least, Osmond and Coop. And they published this in Genetics in 2020, I think. So this was looking at genetic signatures of evolutionary rescues by selective sweeps, just purely genetic systems, of course, but a lot of the work that they did really helped and inspired a lot of the work that I did. So they also have one of the best organized supplementary materials I think I have ever come across. So it's just like worth looking at just for that, if nothing else. This is an excellent article. Okay, so the simulations that I put together, as I said before, they share a lot of the features and the evolutionary dynamics of the analytical model. So it's the same kind of process with the same kind of features. But I've put it into a stochastic simulation framework. So I started out with a Wright-Fisher population of a size about 10,000, and I allowed them to just sort of mutate and reproduce and burn in for about 15,000 generations, which is a lot, but it took a while for the system to settle down, and it's quite stochastic. Then I imposed a detrimental environmental change of magnitude D. So this is the same detriment as we saw in the last model. And then I allowed newborns to mutate at a selected locus. So they could produce beneficial new alleles at a very low rate. And in some populations, so where I was comparing the gene culture case, they were also able to innovate. And we assume that innovation happens at a probably slightly higher is maybe not fair, at a considerably higher rate. So it's much easier to innovate some sort of response to the environmental challenge than it is to mutate a new allele in response to it. After it, I shouldn't say in response to it. It's not quite correct. Okay, so then if the population recovered, so as I said, this is quite like a rare process. So in the vast majority of simulations that we ran, the population went extinct. But if the population had recovered, we then take samples from the population, calculate the average pairwise distance between individuals at specific times. So we're interested in just after the population has recovered, 1,000 generations after it recovered, 2,000 generations, etc., to see how long we could see differences in heterozygosity after the population bottleneck. And the aim here was to compare persistent signatures in pairwise distance or in heterozygosity between gene-only rescues and gene culture rescues. So again, to try and identify if there are persistent diagnostic differences between these two things. So can we tell them apart in genetic data? And then maybe draw some conclusions about the possibility of detecting these in real genetic data. Okay, so these are the results. So across the x-axis here is time, and this is in thousands of generations since recovery. So the 0 here is the burn-in time, so just after the burn-in time. And on the y-axis here is average pairwise distance minus expected heterozygosity. So the expected heterozygosity is just coming straight from Wright and Fisher. This is exactly what we would expect a Wright-Fisher population to do. So this is average pairwise distance minus that. So the red line is basically the analytical expectation we would expect for a neutrally evolving Wright-Fisher population. And you can see that our burn-in, our populations before the detrimental environmental shift happens, line up really nicely there. So the means are the dark lines, or the dark dots, and the lighter dots are just individual simulation runs. So here, the red points are non-cultural populations. So these are just genetic populations. They're not capable of innovating. And these ones are gene culture populations. So these are populations that have genetic alleles and also cultural phenotypes. So after the bottleneck has occurred, so about 1,000 generations after the bottleneck has occurred, you can kind of see there certainly are differences between the way that gene culture populations are adapting to the same environmental challenge and the way that non-cultural populations are adapting. But what you can also see is that these differences decline very rapidly over time. And even if maybe at a short time interval after a population rescue has occurred, you might have a chance of detecting this difference, but this chance, I think, declines extremely rapidly after that. So really, I think what this simulation tells us more than anything is that there are persistent differences here. There certainly are persistent differences, but are there actually persistent and detectable differences? I think that's very questionable, unfortunately. Okay, so I'm not sure how long I have been speaking for, probably not that long because I've talked too quickly, but I'm just going to sum up and then maybe we can have a little bit of discussion. Okay, so what the models showed us was, ultimately genes and culture interfere with one another. And in general, it seems to be fair to say across a large parameter range and in basically more or less every model that we tested, they seem to slow each other down for a variety of very intricate and fun reasons. Both the analytical and the simulation models seem to suggest that gene culture rescues may be much, much more common than gene-only rescues in species where this is possible, of course. And in fact, our, questionably human-like parameter ranges, the probability of a rescue was almost completely determined by the cultural parameters. So it was the cultural parameters that were determining how far up past zero the probability could get. So this was SB, G, and the innovation rate. These are the most important parameters determining what that probability would look like. The models imply that, these gene culture rescues may be more common than gene-only rescues, certainly for, a species like our own, but these also come with shallower population dips, faster recoveries, and so they are considerably harder to detect in data on heterozygosity, so in our simulations at least. So we're working on other methods, but it may be that these kinds of processes are just plainly undetectable from, for example, a single time point modern day genetic data. So it will be too far past when the actual bottleneck occurred. And even if we were quite close to the bottleneck, it might still be quite difficult to detect any differences, or at least to be able to say whether culture was involved in the process or not. So Osmond and Coop that I mentioned earlier suggested that longitudinal genetic data, so for example, some data from before the sweep and some data from after the sweep, for example, might be needed to detect rescues and bottlenecks in other species or the species that they were concerned with. And that may well be the case here. And I think it's probably true that if we also had some cultural data or information about the cultural traits and the cultural processes, transmission processes. So for example, fitness effects or information about the transmission process, we might be able to tell a little bit more about what was happening if we were able to use those two sources of data. So genetic and cultural data together might be more promising. Okay, so that's everything I had to say. Thank you so much for listening. And I'll take any questions or comments that you have. Thank you. I'll stop sharing now, I think so.
Yeah.
But if anyone wants to see any slides again, that's totally fine. I have to find you now. There you are.
Oh, there's a lot to think about. I guess while I'm waiting for other people to, go ahead, Clark. See, you've got a question.
Oh, thanks. Thanks for a great talk. Super interesting and important topic. I guess the very first thing that comes to my mind, I'm mostly interested in humans. I'm concerned about other species, but for, in the human case, is a purely cultural rescue possible? And why, I mean, why wouldn't you simulate something like that as well, I guess I wonder, or can it be simulated? I think about something like a purely cultural rescue in our case, or also a niche construction rescue that might involve culture and environmental change, culture and change in environmental structure without requiring the appearance of a new mutation.
Yes.
Yeah, just curious what your thoughts are on that. Do we have any hope?
An excellent question. I hope we have hope. So we actually so the first rescue simulations that we did appear in a paper that we published in 2019 as like a total side thought. And it was a purely cultural rescue. So we can definitely simulate them.
Oh, good. Okay, great. I'll look
so
for
the
that.
simulations, they're not the best simulations in the world, which is why I wanted to do it again. But they showed that, yes, I mean, you can rescue the population. I think the really big question in that case would be how stably transmitted they are. So I think if I were to sort of do that work again, I would like to investigate, if we were able to transmit cultural traits with perfect fidelity, then yes, I think we could have a purely cultural rescue. But if we have, a slow breakdown of information or something like that, then I think that it might not be as feasible. Also, they have to have extremely high biological fitness benefits. There are tons of cultural traits that have hugely high biological fitness benefits, but it would have to be one of those. So
Gotcha.
maybe rare compared to the full scope of human innovation. I don't know. Yeah, but we have looked at it. Yeah.
Great. Thank you. Thanks.
Dan?
I can wait if there are people in the room who would like to go.
There are not. Everybody attended by Zoom. The one undergrad who came has departed.
Okay. All right. So
sorry, undergrad.
Thank you, Laurel. No, I'm sure it wasn't your presentation. It's just that people have a 1 o'clock class probably.
Well,
and
last week of class, yeah.
I won't take it personally.
Yeah, in fact, your presentation was extremely accessible, and I say that as somebody who has struggled to understand models for decades, and even I could understand it. So so kudos on that. Now the modeler's nightmare is the kibitzer who who sits on the sidelines and says, well, what about this or what about that, right? Because it's very easy to do that versus actually building the model. But it strikes me that there are two perhaps refinements that you might want to consider in this work going forward. So one is, so you showed that the higher the success bias, essentially the greater the buffering or the recovery, right? Which and the rescue, which makes sense. But it seems to me that the success bias shouldn't be constant. It should be positively correlated with the strength of D. Because if people are dying all around me and I see that some people are not, well, now I'm going to pay a lot more attention to that difference than if everyone's sort of doing okay, right? That is, it becomes starker. And so I should imitate the successful more directly. Right? And the second is that the innovation rate obviously matters a lot, the cultural innovation rate. And again, thinking about it psychologically, one might expect that there's something like a U-shaped function between the innovation rate and the strength of D, if you draw analogy with optimal foraging theory, right? So when things are looking a little sketchy, things are a little iffy, I'm not really sure what's, people seem to get sick some, or there isn't as much food as there was or whatever. Now I get conservative and I say, we got to do things the old way, right? And there's actually quite a lot of evidence, including by one of our graduate students, Theo Seymour, about shifts in traditionalism, conservatism as a function of threat, right? And how those things interact with each other, right? So, stick to the tried and true if things are looking a little sketchy, right? But then when things get really bad, now, roll the dice, try something new, right? Because the old way clearly isn't working anymore. And that as things get more and more dangerous, I should be more and more willing to try alternatives. And thus the innovation rate should skyrocket at that point.
That's awesome. I will look up that work on innovation rate because I also am super interested in innovation in general. So that's really interesting. With regard to the success bias, we actually have two models. So one in which it's constant and one in which the success bias is a function not of the frequency of the cultural trait. So not based on not based directly on how many people around me have that cultural trait necessarily, but as a function of SB, so as a function of the biological fitness of the cultural trait itself. So that's kind of a proxy almost.
Okay, just so that I understand, where the biological fitness is itself related to D, because the strength of the selection pressure is going to determine the difference between high fitness and low fitness individuals.
Yes, yeah, that's true.
Yeah, okay. Mm-hmm. Great.
Yeah, so we do have like some, so we have some sort of, so we have kind of thought about that is kind of, I guess, what I want to say. But with regard to the changing innovation rates, I think that's very cool. So thank you for that. I will have a think.
Absolutely.
That.
Very nice work.
Thank you.
You mentioned that you wanted to connect this more with real world data collection. What kinds of data would you love for people to collect? And in what species, ideally? Where do you think would be the best place to look for relevant data for a project like this?
I mean, one of the first things and one of the things that I've been racking my brains about for a long time is what's a very convincing, putative example of possibly a gene culture coevolutionary rescue. If you could pinpoint one in human history, could we think of one convincing one? It's very difficult because I guess like there's no cultural things happen and genetic things happen, and you have to say, well, okay, maybe they're happening together, and can we look for signatures of that? So it's a very difficult question. So I think firstly, the first real world thing that I would really love is a solid example or a solid putative example of something like this happening. The best one, the one that I always keep in my head, is like adaptation to cold. So I mentioned that in the talk, because we know that we have a lot of cultural adaptations to cold, and we also know that a lot of those predate, say, genetic adaptations to cold in Europeans, for example. So we know that. And so we know that human populations have adapted culturally to, for example, European climates before they adapted genetically to European climates, although we did do both. So that's one really nice example. As far as looking for the signatures of these things are concerned, I think the best two parameters that we could try to find or estimate or calculate in some way would be the initial rate of decline of the populations. This would tell us what D is on its own without a cultural trait involved, without an allele involved. That would be really helpful. And also then the SB, so the selective advantage conferred to individuals with the cultural trait. And I think with those two things and all of that genetic data, I think we could do more. as far as detection was concerned. Those would be my ideal kind of points.
Would there be anything about the pandemic, mask wearing and all this other sort of stuff that's come along with it that would be useful?
I don't know.
There are a lot of cultural traits that it seems like a lot is changing culturally right now, and there's a lot of mortality. I don't know if any of that kind of stuff.
Yeah, that's true, actually. I hadn't really thought about leveraging. I mean, that sounds like a terrible thing to say.
Brutal, but I don't know. Maybe data that showed it might encourage people to modify behavior quickly or something.
Yeah. I mean, well, that's for sure. Yeah, I mean, I hadn't thought about it, but maybe that's a really good point. And mortality was high. So,
Brian.
Hi, Laurel. That was a really enjoyable talk, and everything
Thank
Dan and Clark and Susan have said, I just want to second that. It was really well presented, and it was great. And I guess I wanted to ask about some of the details of your modeling approaches and how they might handle the following kind of cultural evolutionary process, which would be, at least in your schematic description of your simulation, it looked like there was this lethal shock that occurred at a fixed point, and I assume affects all individuals in your simulation at that moment in terms of changes their environment. And I was wondering, because cultural evolution, cultural transmission really has this neat property of horizontal transmission, especially to people in your cultural group who might not be in the environment that some sort of shock or stimulus is occurring in, but the information can sort of disperse across through storytelling, so that in effect what could happen is that these environmental shocks might be, some subset of individuals could be aware of this shock at some point over the horizon, a place beyond their personal individual experience and exposure, and how if you built in this sort of possibility for a more lagged exposure, and you could even have instances of individuals with cultural traits that re-equip them for some lethal shock that they can anticipate, like the cold example that you mentioned. And this, I don't know, I just thought I'd raise that issue. And at the scale that you're doing your analyses, I'm not sure it would have any real effect, but it's just something that occurs to me as something that goes on. People are able to tell stories and sort of prefigure what kinds of threats they're going to be facing through culture.
I mean, that's a really good point. So I think there's kind of two things that stand out to me about your comments. So the first is whether there could be individuals in the population who already have cultural knowledge or cultural traits that are important or pertain in some way to the environmental shock. So we looked a little bit at this. So one of the papers that we published was trying to look at what's the difference between adapting culturally using standing variation. So variation that already exists or has come into the population some time ago or persists at like a low level in the population and then becomes relevant in some way, versus waiting for innovations to arise from de novo kind of innovations. This is sort of equivalent to kind of de novo mutations. So these are all in the genetic models already, but we tried to do a cultural version of that. So we found, of course, that adapting from standing variation is much faster and it's much more efficient. And it sort of also relies on what was in kind of the second part of your comment, which is foresight. So what we kind of looked at there was that there's a little bit of a trade-off, right? So if you have experience an environmental shock and you wait to experience the environmental shock and then you innovate something in response to it, and the probability that you have sort of a matching cultural trait to that environmental shock is higher than if you're relying on some kind of foresight. So you're sort of guessing what's going to happen in the future. But I have this standing variation and I've done a little bit of guessing, and this kind of thing. So there's some trade-off between those two things. You get better innovations if you wait, but you have much faster adaptation if you use foresight and have those traits sort of ticking over in the population early on. So we've done a little bit of modeling in the context of selective sweeps more than the rescues that sort of pertain to that. But I mean, you're right in that it would be interesting, I think, in this model as well. That's a good point.
Thanks. That's really interesting.
Feeling I'll have more questions once I've had a chance to think about it more. So,
I guess I could follow that up, because you have a wealth of other models that it seems like you've been exploring all these ideas. So I was, when I think about your example of a putative genetic rescue based on or a selective sweep or some combination of genetic and cultural adaptations to cold, strikes me as one kind of form of cultural adaptation would be operating in that kind of a context where you have this like macro ecological process. Cold is something that every individual can perceive. Even in the warmest climates, people have an awareness of temperature differences and how to adjust to them. So it seems like of all the possible kinds of environmental shocks that you could come to it with pre-existing cultural, wherewithal, something at that scale would be a good scenario for drawing on standing variation. But then as a completely different scenario, when a pathogen enters a population that you've had no exposure to, or maybe you have had exposure to it, or some people have in the past, but even understanding the mechanisms of the disease or interventions that could mediate its lethality are way beyond human comprehension. Let's just say, for this, I'm just saying this because of the COVID example, like how should we culturally adapt to COVID? I don't know if we really know. That's even with as much, tools and thought brought to bear on it. So I just want to mention this dimension. I know it's just yet another complication, but like these kinds of dimensions of certain, I mention this because you could imagine certain kinds of environmental shocks being less likely to be handled on the cultural side, and if they are handled, only to be done so at the genetic side. So that might somehow constrain the search that you would do in the genome. I mean, presumes a lot that we know what allelic changes would have, what kinds of impacts it would have on phenotype. But I'm just thinking, like, there could be some coarse-grained analysis at that level, trying to see like what are the least probable culturally solved problems, and then expecting that to be more likely to be found in genetic signatures of selection. I don't know if that makes any sense. But.
Yeah, no, that's a really good point, that you can you could maybe pull apart where you would be likely to adapt behaviorally or culturally before you adapt genetically, and where you would be likely to just fail to adapt culturally. I'm sure there are tons of scenarios where that's the case as well. That's probably like a really good kind of first pass at finding examples. Yeah.
Mark?
I have a question that's maybe way more a little more difficult, because it's going outside of the box of what you've talked about. But these days we have all of this discussion of existential threats to humanity. It seems like almost all of them are endogenous. So in your model you have an exogenous thing happen, right? Like I would guess it's something like a meteor hits the Earth, or the appearance of a new virus or something, right? But arguably climate change is not entirely exogenously caused. If ChatGPT ends up killing us all, it's our own fault. So, I mean, I guess I'm wondering, and I guess for some kind of risk like that, I would assume that genetic adaptation could be part of the solution to, obviously that's a possible thing that could happen in response to an endogenously caused event. But I guess from a modeling level, I don't do much modeling myself, but I'm just trying to wrap my head around, how might you model that kind of thing? It seems like it might be like a Lotka Volterra type thing or something. I don't know. I mean, how would you model like that kind of thing where you don't just assume an event that occurs exogenously? Do you have any ideas there, or yeah?
Yeah, I mean, I think, so firstly, your comment is really interesting because a lot of these sort of canonical examples of gene culture coevolution are exactly what you're describing. So lactase persistence, the environmental change that changed the selection pressure on the lactase persistence genes were cultural. And so there's this feedback system. The same thing with the sickle cell example is exactly the same. So these are all endogenous processes. And I was actually thinking about that today because I was thinking about this idea of examples, and I was thinking, these sort of canonical examples of gene culture coevolution just don't quite fit with what we're trying to model here. It's kind of a different phenomenon. But in answer to the question of how I would model that, I also model ecological inheritance. So I work a lot on non-genetic inheritance because I'm interested in human evolution. So I do a lot of niche construction models. So some audiences like this and some audiences despise it viscerally. So I apologize if you fall into the latter category. But I would model it using a niche construction or cultural niche construction framework, I think. That would be my first instinct, because this is, so I think, and also a lot of the, maybe not the cultural niche construction models, but the niche construction models in general tend to separate a niche constructing locus from a niche constructed one. And that would fit quite nicely with that. So you would say like one cultural trait is affecting the environment of the other cultural trait, which is then maybe sweeping to fixation or something like that. So I think that would be my first instinct for how I would do something like that.
Yeah, I mean, that matches my intuition as well. I think niche construction is an excellent way of thinking about these kinds of problems that we're facing now. I wonder if you might have, I also, when I try to think of like, what is the sort of category similarity of the self-produced existential threats that exist to humanity right now? A lot of them seem to result, I don't know, but my intuition is they result from runaway type of processes, maybe, or I don't know if that's not quite the right word, right? But when you think about something like ChatGPT, I don't know if that's an existential threat at all, but imagine that it is, right? I mean, it's because of a competitive process, and that people who talk about the development of the software basically say, the problem is there's no mechanism to stop people from continuing to make it better, better and better from its own lights, right? Same with the kind of runaway processes that are involved with environmental destruction. Even if some of us, want to fight very hard against it, there's, the problem is basically actors that generate it anyway, and it's through a competitive process that's actually in some perverse way selecting for the agents and the systems going more towards destruction, right? And so it seems like that somehow seems also like maybe to have a different could have a different dynamic than when we talk about like lactase persistence evolution or something like that, because it wasn't exactly, I don't know, the dynamics seem different when you have the threat resulting internally from a competitive dynamic. And then you kind of wonder, it would be interesting to model the question of like, how does a population recover from threats that are generated by the selection structure, within their own population to produce these selfish, maybe you could argue they're selfish things, right? And so it's a cooperation problem or a tragedy of commons type problem. I don't know.
Maybe. I mean, it sort of touches on this model that we published. I mean, it's such a long time ago now. Oh my God. During my PhD, which was this kind of runaway cultural niche construction paper. And it looked at how we could evolve costly things. So, I mean, essentially detrimental things if they helped us to niche construct more. So we were thinking about brains, for example. So it helped us to, create more culture. And so we kind of kept getting, bigger brains, and they're more costly and whatever, but we could compensate for it. So these kind of runaway processes are actually like very cool to model and can be quite difficult to stop. Yeah, but there's definitely a framework there, I think, to do that. It was very interesting.
Yeah. Thank you.
Dan?
So a couple of thoughts following up on Clark's and Susan's and to some extent Brian's remarks. First, I should say with a big caveat that I'm skeptical that that any sort of observable contemporary human events are going to be amenable to the degree of simplification that is necessary for translation into a model, because there's so much going on. But having said that, I'm trying to be helpful. First, in response to Clark's idea of basically an internally generated threat, I wonder whether Melanesian mortuary cannibalism in the Highland PNG with endemic Koru prion disease offers any possible avenues. I don't know whether there's any whether it's possible to evolve resistance to prion diseases because they're so different than pathogens per se. But this is entirely an internally generated institution, presumably generated by some of the kinds of prestige competition processes that Clark was referencing, although not in the scale or the degree of competition in a marketplace that leads to cutting down the Amazon or something like that. But there's a fairly well-studied, fairly localized. It's clear what all the relevant variables are. So, like I said, I don't know if there is resistance to prion diseases, but that might be one possibility. In regard to Susan's suggestion about COVID, I'm quite skeptical of this for many of its utility for your purposes for many reasons. One just being that the mortality rates are highest in post-reproductive age or prime reproductive age individuals, and so selection, despite high mortality rates, selection is going to be weaker there by definition. And I wonder whether a better case and relevant to Brian's sort of opacity point, right, which is can people solve the problem, right? Like it's very hard for people to wrap their heads around the idea that somebody else's exhalation could kill them, right? That doesn't fit with their evolved intuitions about disease transmission, right? But I wonder whether a case that might be where there might be the variables are more discrete. It is directly relevant to reproductive age individuals and in fact the act of reproduction would be HIV, particularly in sub-Saharan Africa. So the APO BEC8, is it APO BEC3H? I think it is. I believe is an allele which has been lost in lots of populations, but remains more frequent in African populations than other populations. It conveys resistance to retroviruses. In the absence of medical intervention, AIDS is always lethal. It's hitting reproductive age individuals, who are sexually active, and the cultural interventions that protect against it are, both, readily understood and not as counterintuitive as, for example, COVID transmission prevention is, right? Because I think people recognize sexual behavior as an opportunity for disease transmission more easily than they do other things, because they're, historically over human evolution, there have always been sexually transmitted diseases, right? And bodily fluids are a strong stimulus for an evolved disease avoidance psychology and so on, right? So I don't know whether there is evidence about resistance, evolution of resistance to HIV in African populations, but I do know that tragically mortality rates even now continue to be fairly high in some areas. So that might be, more amenable to this question than something like COVID.
Thank you so much. That's so cool. That is a really nice example, which I will definitely look into. The Koru example, you just made my night, because that is one of my absolute favorite examples of gene culture coevolution ever, because there was a 2009 paper, I think, which looked at the spread of resistance alleles to Kuru in Papua New Guinea, and correlated it with the intensity of the cultural practice. And they were able to find the highest frequency of the alleles in the places where there was the highest sort of frequency of practice of funerary feasts. And so that's like, I think, a really, really cool example. I'm not sure, I mean, this is probably another one kind of like the sickle cell, where the cultural practice is itself causing the environmental change. So it's kind of maybe more akin to those ones, but it is a super cool example. I mean, there are cultural evolutionary models of Kuru, even in, I think, Kvalsworth and Feldman's book as well. I think they have one like hidden in the back there somewhere. That's quite a cool model as well. But yes, but thank you so much. I will actually have a look at that HIV resistance allele. That might be a very cool example. Thank you.
Neat. Thanks.
Any more questions from the Zoom audience? Looks like there aren't, so we can let you go to bed. I see it's now dark out there.
It is the sunset in the time it took me to talk about that.
Yeah, well, thank you so much for staying up late at the office to talk to us, and it's really
no problem. I'm glad I finally got to actually talk to you guys, so I'm really pleased. Thank you, Molly.
All right, well, thank you, Laurel, and we'll be in touch. Hopefully visit soon.
Okay. Cool. Yes, hopefully see you soon, Susan.
Yeah.
Thank you guys so much.
Thank you.
Bye.